speaker for 2024 Gogec is Professor Tae Seok Moon. And he's a longtime supporter of Gogec, so we really appreciate his help in helping get the conference off the ground and organizing it. And he's received a large number of awards and organizes an awesome speaker series called Sinbis, which I recommend people check out if they want to see really interesting talks about a variety of different topics within the SynBio field. And today he'll be speaking about systems in synthetic biology, constructing smart and programmable microbes to address global problems, and has a focus on education in addition to synthetic biology stuff. So go ahead.
Thank you, Alex. This is my great honor to speak again. I think the 3 times in a row in every single, competition and this meeting. As I mentioned earlier, this is my favorite, the best, meeting and competition or conference ever to me, because young people organize, by spending your time, devoting your time to this wonderful platform for free education. So I'm again, super delighted to give my talk and, to give, some idea about me. I'm the Engineering Biology Research Consortium Council member, also the SYMBIS Seminar Series Chair, and I hope I could talk about that seminar series at the end. So last year I formed a company after I got the Langer Prize. If you don't know who is the Bob Langer is probably the most famous influential engineer in history or in current life. He's an MIT professor. He's also founder of the Moderna. He has a lot of company, he found or helped. And then he is the legendary, I believe. And then he started a prize series a few years ago. And then I got this award. And then I should form a company and I call it Moonshot Bio. So I'm just showing the conflict of interest slide right now. And then I believe the deadline for this year, probably this is the oldest slide coming soon or already passed. So if you wanted to submit, I mean, that is one thing you could try. That's up to $100,000 you can use for your great idea for the future. So let me start with my dream. My dream is engineering biology, and it's our ancestor dream of flying in the sky. But instead of creating birds or transforming themselves into birds, they analyze, of course, birds and design wings and propellers and assemble them together to make airplanes. I want to do exactly the same thing. Instead of creating new creature, I want to analyze existing cell and design synthetic DNA and assemble them together to make engineered cell that can be used for biofuel production, chemical material production, some diagnostic, therapeutic application, and all the way to bioremediation and some other application, including creating plastic eating bacteria or carbon capture bacteria, etc. And obviously we call it synthetic biology. Currently, I have more than 10 projects to achieve those kind of big, ambitious goals. I'm not going to talk about all the projects I'm currently working with my student and postdoc. And then I'm also constantly trying to hire many postdoc and actually postdoc mostly. So if you're interested, please contact me at my Gmail address in this slide. So the question I ask, when I start my position, 2012, now almost 12 years ago, the question was, what if we can combine genetic sensors and genetic circuits and so-called actuator, that's basically function, cellular function, in a human-defined manner. If that is possible, we can make bacteria work for us from tumor-killing bacteria, basically sense tumors and only inject some killing agent into the tumor. In that way, you differentiate between tumor cell and normal human cell. And then we call it tumor-killing bacteria, all the way to making plastic-eating bacteria. That means bacteria potentially deploy into the environment with some safety circuit. We call it kill switch. And then bacteria move around. And then when they sense plastic, they basically secrete some enzyme that degrade plastic and then just clean up. And then once that cleaned up, basically bacteria also self-destruct for the bio-containment goal. And then there are multiple different applications you could imagine, and basically your imagination is basically the limit. But to do so, we need to kind of combine these three things together. So for the first topic for today, I want to talk about one of my biggest passion project. This is basically a 12-year project at this moment, and I call it smart probiotic construction or engineering. The idea is basically the same. We use some sensor that basically detects some disease or disease marker. And then once that is detected, bacteria do something, generating some color or even generating some molecule that basically cure the disease. And that is basically function or actuation. So obviously to do so, we need to have lots of sensor first that basically detect those kind of medically relevant signals. But unfortunately, when I start my position 12 years ago, we did not have many sensors except some sensor people used to use, including IPTG sensor that responds to small molecule IPTG or lactose. That's a very important, useful sensor to study gene expression or overexpression of some specific gene, but that's not that practically, useful because that is only response to IPTG or lactose, unless you wanted to study or develop some therapeutic method to treat lactose intolerance. So our lab basically start to develop some more application relevant sensor by thinking about what happened to the probiotic when you take yogurt. So think about, when you take yogurt, what happened to that probiotic inside the yogurt. Basically, when you take yogurt, bacteria in the yogurt basically go through your body. First, your stomach, that's basically low pH, around 1 or 2. And then when that bacteria all the way go to your gut, that's basically anaerobic condition, no oxygen in your gut. And then once your bacteria come out of your body, basically temperature shift from 37 Celsius, that's a body temperature, and outside temperature is typically lower than 37. At night, at least, even in the hot summer of Africa, temperature probably go down around 30 Celsius a certain day, not always. But when that happens, bacteria experience temperature downshift from 37 to some temperature like 30 degrees. So to develop such sensor, we kind of use synthetic biology technique all the way from developing temperature sensor, that's the upper left corner, and then oxygen sensor and pH sensor. We also recently developed one sensor that differentiates between potentially large intestine and small intestine by focusing on the one chemical called propionate, because propionate only exists in the large intestine but not small intestine. In that way, if you wanted to deliver some molecule only in large intestine for the therapeutic purpose, you can use this sensor, and then this sensor basically allow no expression during the passage of this probiotic, but when they alive large intestine, now the therapeutic protein basically produced. My lab also very interested in pollution or climate crisis. And then one of the pollution we are looking at for many years, almost 10 years, is the phenol or aromatic compound. And that this sensor is one of the example, specifically detecting phenol, but not other aromatic compound. That's basically I'm showing in the bottom left corner. So we develop a lot of sensor. This is kind of old slide with all the list of the sensor we develop. And then you can just check out all the papers in this slide. But you may say this is not medically relevant sensor because that is not disease marker detection. So during pandemic, actually we several years ago we did extensive literature search to find what kind of sensor is available. I think that's 2017-ish. And then during pandemic, we basically summarize our findings from the literature search and then published 2 papers about available sensor. And unfortunately, this table at the time is probably the only available useful sensor you could use. That is for the medical application. But as you can imagine, not that many sensors available here. Some interesting sensor I want to tell you today is one sensor developed by MIT professor at the time, Tim Nu. They basically use some protein that bind to heme, that's a blood component. And then once this binding happen, this sensor basically generate luminescent signal, and that luminescent signal converted into wireless signal that is detected by the machine outside. In that way, if you have the internal bleeding, without using endoscopy or some invasive method to detect bleeding inside, you now need to only take one pill. That pill contain device and probiotic, and that combination basically generating wireless signal, and then outside device basically detecting the bleeding inside. So after realizing we still need to have many sensors, that's around 2017, I want to focus on developing new sensor. And then I found one interesting paper that is talking about neurochemical or neurotransmitters, in fact, also found in your gut, in your intestine, rather than you believe neurochemical actually produced in the your brain, but in fact, neurochemical also found in your gut. At the same time, interesting finding was one of the most famous neurochemicals called serotonin. The serotonin actually produced by your gut cell, your intestine cell, more than 90%, and only less than 10% is kind of produced by your brain cell. So after I see this kind of paper, I was so fascinated because I believe I'm just human and then controlling my mood, happiness, everything by brain. But that finding kind of tell me not necessarily that's the case, because your gut cell also influence your happiness and mood, because of serotonin basically control happiness and mood, etc. Another interesting point I just found was your bacteria cell in your body, in your gut, in your skin, in your tongue, all your bacteria, total number of bacteria cell actually much larger than your human cell. So that means you, I believe you are a human being, but yourself defined by your total number of human cell plus much more cell you carrying, that's a bacterial cell in your body. And then when you travel, you exchange the bacteria, and then that bacteria potentially influence not only because of the disease, but also the bacteria in your gut or in your skin somehow generating some interesting chemical or influence potentially your brain. Let me tell you one example. The serotonin produced by the gut cell of your body, at the same time, some bacteria even metabolize serotonin in your gut, degrading or synthesizing, in that way potentially influence your brain function or your happiness level or your mood. So after thinking about this kind of problem, this fascinating problem, I realized I probably need to work on this project to develop neurochemical sensor. But one thing I want to really do in the future when I start this project, I wanted to make yogurt you take in the morning, or sometimes 2 times a week or whatever. I want to develop yogurt that contain probiotic, and the probiotic basically detect some neurochemical. And then if, for example, let's say, we develop serotonin sensor. And then if the serotonin level is between 30 to 100, let's say that is the right level, make you happy. And then, for example, today you just take my yogurt I develop, and then you look at your poop. I'm sorry, I'm talking about poop before lunchtime or after dinner time, depending on where you live. But let's say you take my yogurt, you look at your poop, and then let's say your poop color is now green because the bacteria in the yogurt basically detect serotonin, and then somehow your serotonin level is between 30 and 100. In that way, bacteria start to produce green color pigment. And then when you look at your poop, you're basically seeing green color. That's because of my probiotic. That means today is the happy day because your serotonin level is just right. But life is not that simple, sometimes good, sometimes bad. Unfortunately, tomorrow, when you see your poop, now your poop color is red. That means your probiotic bacteria now detect too much serotonin or too little serotonin. In that way, bacteria start to produce red pigment. So that's kind of, you may think dream or movie stuff, but that is actually possible and people try to do so. Not necessarily for the neurochemical, but some other disease marker. But why we are stuck there? I mean, we can go one step further. So imagine, temperature control in your room if you have the air conditioner or heater. If the temperature set is something like 25 Celsius, and then somehow temperature fluctuate, and then let's say temperature go above 25, And then when that happened, temperature controller, thermometer, basically thermostat, basically kick in air conditioner to make the temperature go down. If temperature too low, now heater start to heat it up the room. In that way, temperature go back to the 25. We can do the similar thing by using bacteria, because as I mentioned, there are multiple pathway or enzyme that degrade serotonin or synthesize serotonin. If you connect your sensor with the enzyme, depending on the serotonin concentration, if the serotonin level is too high, you could ask bacteria to produce enzyme that degrades serotonin. If your serotonin level is too low, bacteria could produce enzyme that synthesize serotonin. In that way, your serotonin level is just within the right level, that's between 30 and 100. And then I already have my company's commercial punchline. That is basically, I would say, do you want to be happy, take my yogurt and you will be always happy. So that's my dream kind of project, but that's not gonna happen tomorrow because there are many, many, many challenging problems I need to overcome or solve. First problem is, of course, how to make sensor that is specific enough to only one chemical, not the other chemical. Let me give that example using this simple two chemical, neurochemical over here. One of them is the PEA, phenylethylamine. I call it PEA. The other is the tyramine and TIRA. The only difference between these two compounds is basically one compound does not have oxygen in the aromatic ring. The other has the oxygen. But the consequence of this different chemical is hugely different. One is basically related to psychological disorders. The other is basically related to some food poisoning or some other thing. So this is a very important thing. And if your sensor is only detecting all of the similar chemical, that's typically the case, the natural sensor, the chance are, if you have the sensor like that, your doctor may say 50-50 chance of you have some psychological disorder or just food poisoning. So misdiagnosis chance. So we wanted to develop the sensor that's specific enough to only detect one chemical. But as you can imagine or see from this slide, there are multiple different chemical, but very structurally similar, including dopamine has 2 oxygen and tryptamine also a little bit different structure, but still have the amine group and aromatic group over here. And then developing sensor that is specific enough is not that easy thing to do. In fact, we spent more than 2 years, and then during this 2 years, we cannot get any sensor specific enough to sense those neurochemical. And then we are kind of start to worry about at the time because the entire project is only a 3-year project. But when we test all the literature reported sensor, none of them actually worked, unfortunately. So we tried to come up with new ideas that basically now we call it dual knob sensing mechanism. This is kind of a little bit unique because typical sensor, we basically use some protein, typically transcription factor. That factor basically bind to the small molecule. And then that binding basically change the conformation of that transcription factor that trigger binding between DNA and protein complex with ligand that basically generate, in this case, GFP signal. But we realized the real ligand for this case is not the amine, that's our neurochemical, but the conversion product of amine catalyzed by the tin A, that's an enzyme that converting amine into aldehyde. So you may say this is not necessarily amine sensor, this is an aldehyde sensor, but luckily for us, aldehyde is unstable. That means live short only for a short time. And then in the diet and then in the body, this aldehyde is not, detectable. So we can still say this is an amine sensor because the TNA conversion reaction is very quickly, happening. But the problem at that time is still we basically have sensor, in this case, VRR, responding to all four chemical I just show here. So that's not specific at all. So, and then we got, basically, we got the COVID pandemic at the time, and then the entire lab shut down. Of course, all the university shut down for several months. And then during that time, we just constantly worrying about because we only have 1 more year, but luckily we have the 1 more year extension from the DoD, Defense Department in US. And then during that time, one of my students, Matt, approached me by sending email saying he wanted to learn new algorithm, computational algorithm, including AlphaFold or Rosetta. That's basically protein prediction, structure prediction algorithm. And then you can also use those kind of prediction algorithm for the ligand protein docking experiment. Essentially, you could kind of simulate those binding interaction. And then you could kind of play what kind of residue in the protein you could change to make this sensing more specific. So basically, he asked, can I, learn this kind of program? And then to me, nothing to lose. So I just said, why not? Just go ahead. And then several weeks later, he sent another email saying, Professor, I learned how to use this algorithm or programs. And I was so delighted. And then several weeks later, he predict 5 different residue in the protein. And then we just need to change each residue to 20 different amino acid or 19 different amino acid. That means we need to only screen 100 different variant of sensor compared to 10 to the 7 or 8 different variant we screen when we use only aeroprone PCR-based directed evolution before, but we never make that work because there are many false positive or false negative, and that always giving us some cheater or not functioning sensor. But after we screen 100 of them, we identify many successful functioning sensor, including FEAR A81L. You can see that graph on the right side that is only respond to PEA, but not other chemicals. So to give some short summary, rather than showing all the data we generated, this is the default neurochemical sensor specific to only one neurochemical, and also giving some biological output. Important thing, I want to say, we still do not have VRR crystal structure, but this prediction algorithm basically give us some insight, the structure of the VOR, potentially enabling future development of new sensors. Actually, we currently develop lots of neurochemical sensor, more sensor for other chemical, and then we're about to submit the new paper in near future. And then recently, actually last December, we did more extensive literature search to find what kind of sensor developed last 3 more years. And then we are very glad because now we have the more sensor we could use. And then if you're interested in sensor development, this extensive, comprehensive review paper will be very useful. Actually, I think this is the longest paper I ever written with almost, I think, 20 pages with 235 references. So this is the first part of my talk, and then let me just kind of talk about the second part. That's all about bio-containment. You may say, Professor, or Dr. Moon, that's cool. I mean, you could cure disease or make us happy all the time. But at the end of the day, this is a genetically engineered microbes or GMO. I call it GEM, GEM. And that potentially release into the environment because, bacteria come out of your body with your fecal matter. So to ensure the safety, to the environment or potentially to the human, we wanted to make sure bio-containment is ensured. So we basically try to develop this kill switch. Basically, the once bacteria accomplish their mission, in this case, curing the disease or make us happy, once the bacteria come out of your body, and bacteria should die. So to do so, to make the long story short, we basically use multiple different mechanisms, but we finally settled down with one mechanism, that is the CRISPR system. As you know, CRISPR is basically seizure that cut the DNA. But if you target your genome, you basically cut genome. If you're dealing with the bacteria, most of the bacteria do not have so-called non-homologous end-joining mechanism. That means once you cut DNA, bacteria in theory should die. This mechanism probably not going to work with the eukaryotic system because eukaryotic system has the non-homologous end-joining mechanism. That means stitching DNA together by introducing some error typically, but still eukaryotic system may survive with some mutation in the DNA. But this is not easy problem to tackle because we are talking about killing bacteria. And as you know, no bacteria want to die. No human want to die. No bacteria want to die. So the first version of this kill switch is basically showing this data over here. And I explained this data a bit slowly. The y-axis in the bar graph is basically colony forming unit per ml. And that is basically start around 10 to the 8 cell per ml of culture. And then white bar or gray bar is basically without induction. Red one is with induction. The first column is the most important data set because that is the strain without kill switch. And as you can see, there's no number change between no induction and with induction. The rest of the 6th column is basically kill switch containing strain. You now see 10 to the 4th reduction in terms of number of the cell. except one of the guide RNA, we target one of the region in the genome. So 10 to the 4 cell reduction or 10,000 fold reduction is a lot of killing, but this is not necessarily acceptable because even one cell remain after this induction, that one cell may become 2 and 2 become 4, and then that may dominate entire population. And then that is something, we need to prevent if this bacteria come out of your body. But always, some, life system may find some way to survive. And then that's typically because of the mutations or evolution. So I'm showing one of the typical example, when we try to improve the killing efficiency. But the first version of kill switch, basically we see a lot of deletion in the promoter region that basically killing the Cas9 expression cassette. In that way, we kind of seeing one escapee that become 2 and 2 become 4 and then dominate the population, giving you 10 to the 4th cell per ml. So we try multiple different things, including knocking out error-prone polymerase, DNA polymerase. In that way, reduce mutation rate. We also try multiple different things. But at the end of the day, what I found after visiting hospital for my, doctor's, appointment, and then I realized what happened at the time if we lose electricity in the hospital when I waiting for 2 hours for my doctors. And then my answer was, doctor still able to operate or surgery because developed country in the hospital, they always has backup generator. After thinking about that random thing in the hospital, I realized, why not? We can have multiple kill switch. In that way, mutation destroy one of the kill switch. You can make other kill switch still working as a backup. In that way, you should have all the kill switch destroyed by the mutation at the same time. In that way, some bacteria potentially escape. So to make the long story short, actually this is a 7 years project with a lot of failure and success. My student Austin never give up and then finally make everything working perfectly. And this is the basically mouse data. Here, the green and gray color or white color is basically control. And as you can see, your bacteria probiotic without kill switch stay the same number as at the time 0. But if your bacteria has the kill switch, now bacteria start to die. And then after 48 hours, there's no survivor at all when we use the mouse experiment. And then actually that is actually when you use the ATC, because we wanted to use small chemical anhydrotetracycline if bacteria we engineer or GMO or GEM, if the bacteria somehow become bad guy in your gut, we only to target, only kill that bacteria by using chemical without touching other bacteria in the gut, because using antibiotic typically result in bad consequence, including some bowel disease. But real biocontainment should use some environmental signal to differentiate between the body temperature and outside temperature. And as I implied earlier, we can use temperature sensor to differentiate between 37 degree and or the 30 degree or below. So we used our developed temperature sensor in this case, and then we basically show the same thing using mouse model, and then we demonstrate complete elimination of bacteria if bacteria come out of your body or mouse body. So where we are going with this technology, we are currently developing many different things, including plastic-eating bacteria. Once bacteria eat all the plastic, clean up our environment, bacteria should die. We are developing some kill switch in photosynthetic bacteria that typically deploy in the open pond, but you may heard algae bloom that destroy ecosystem. If that happens, we can add some chemical. That chemical basically only target GMO, probiotic, the prokaryotic algae, and then we can basically kill only that guy if they grow too much. We can use this kill switch if we wanted to deploy some bacteria to extract rare earth element, because rare earth element is very in high demand, but that's a rare earth metal, and then you should use some technology to extract that rare amount of the metal in the environment. But at the end of the day, we are basically deploying the GMO into the environment. We should have some way to make them die once they extract all the rare earth element. So this is kind of the project we're currently working on to implement kill switch as broadly as possible. One recent example, we just almost finished and then about to submit, we actually developed kill switch, and the idea is basically this bacteria cleanup. Phenol is the toxic chemical that cause cancer or some other disease. But once bacteria eat all the phenol, bacteria basically self-destruct. In that way, you clean up environment, at the same time, your bacteria also die to make sure the bio-containment. So where we are going with this one? So my biggest vision in this field is so-called microbiome engineering. So if you are working on metabolic engineering, you probably, read a lot of paper talking about one bacterium converting one biomass or feedstock in one bioreactor to make the one product. That was paradigm in metabolic engineering more than 30 years. But why we limit ourselves to this one situation rather than thinking bigger? So recently, I published one opinion paper in Trend in Biotechnology. Now I see it, for example, entire soil or water or entire planet as huge bioreactor. And in that bioreactor, you may have photosynthetic bacteria that capture CO2 or nitrogen-fixing bacteria that capture nitrogen and making ammonium. In that way, you don't need to worry about chemical nitrogen fertilizer. As you know, that nitrogen fertilizer in developed country, we use too much. In that way, there are lots of other problems like algae bloom or etc. But country in the developing country like Africa and other country, they do not use enough nitrogen fertilizer. In that way, crop yield is very, very low, creating food inequality. You can use some plastic-eating bacteria. I'm gonna talk about that in a minute. You could use plastic-eating bacteria to potentially converting plastic or waste into high-value chemical or material. In that way, sustainably produce some bioproduct. To do so, we recently developed a tools that is basically engineer microbiota, at the strain or species level. Now we demonstrated we could kill only one bacterium out of, in theory, billion different bacteria. And then you could use this tool to target only one pathogen in your gut or in your soil. In that way, only kill plant pathogen without touching or affecting other bacteria, including beneficial bacteria. We can also use the same thing to integrate some antibiotic resistant gene only to the target bacterium. And then we do selection based on the antibiotics. In that way, you could facilitate isolation of beneficial bacteria from the environment. Another thing I want to talk about is antibiotic resistant, spread problem. People basically blame clinic or farmers because farmer actually the user of antibiotic. Actually, they use the most of the antibiotic to make pig, cow healthy, but that potentially generate superbug that resistant to many different antibiotics. Also clinic, we use a lot of antibiotics in clinic. But what I want to say, have you thought about when you're doing cloning, should we destroy the DNA you made? I'm talking about plasmid that typically contain antibiotic resistant gene in it. We kill bacteria after experiment because that's a government regulation, but nobody so far destroyed the DNA because there's no regulation about DNA. But more than several decades, we release this killed bacteria into the sink, and then that contained plasmid that has antibiotic resistant gene, and then we are culprit. We are also the people we should blame because we potentially contribute to antibiotic resistant spread because that DNA basically transport to other bacteria, including pathogen, making them resistant to antibiotics. So to solve this problem, we recently published one paper that is basically use essential gene and auxotrophy. In that way, now you have the routine way to use antibiotic resistant gene-free plasmid system. You don't need to worry about developing this one because the only thing you need to do is asking me this strain for the cloning and then order plasmid from Adagene. I believe we already shipped our plasmid to Adagene. You can just order that plasmid and then you can use the routine cloning without using antibiotics. In that way, you potentially save some money for the buying antibiotics and then potentially minimize antibiotic resistance spread. Because of time, I probably skipped this one, but basically we show this plasmid, maintain up to 1 month without using antibiotics. And then that's like, using the mouse model, we also show that it can maintain for a while. So I will spend, a couple of more minutes to talk about, briefly about my metabolic engineering effort. The main idea is basically plastic upcycling. And, I've skipped all the slide for now. But bottom line is plastic is, huge problem in the world. But now we develop making the process using the plastic, including the PET. And then we use our scissor, the physical scissor, to chop up the plastic bottle. And then we use the chemical method to convert that into a smaller monomer. And then we use basically bacteria to convert that toxic intermediate into lycopene. We focus on lycopene production because currently lycopene is produced by extracting lycopene from tomato. But my dad always tell me, don't play with your food. So I don't want to play with my food, because at the same time, when people play with food to extract this high value lycopene that's up to several $100 per kilogram, many people die in Africa and some developing countries because they don't have food. Not necessarily tomato, but conceptually, using food to extract chemical is not acceptable practice. And I want to disrupt this practice by developing economically viable process to make the lycopene out of the plastic waste. Currently, we're only making 1 milligram per liter, but we recently improved the titer more than threefold. Now we are using multiple different plastic, not only PET, but polyethylene, polypropylene, polystyrene, and also the PET, the mixture of them. And then we actually preparing another more than 2 papers to talk about how to convert mixture of plastic waste, PE, PP, PS, PET, into high value chemical or some bulk chemical in the future. So with that, I will just talk about my education, free education effort. That is basically, I call it Synthetic Biology Young Speaker Series to nurture young, rising star, including many speakers in this Gogec conference who already gave a talk, including, Duhan earlier before, my talk. Also the Friday and Saturday, there are many people spoke during this seminar series. I basically pair one pioneer, including Frances Arnold. She is the Nobel Prize winner. But every week, I'm running this seminar series. And then this is available for free through YouTube channel. So if you are interested in learning more about synthetic biology, you could type my name or Synbis, and then you will see more than, I think now, 100 video talking about synthetic biology in a different area of topics. So this is all the sponsors who support the seminar series, including almost all US funders, including ONR, NSF, USDA, NIH, at NASA, etc. Also big journal like Nature and Cell Journal editors, 9 of them supporting by speaking in this seminar series. And of course, the US government, including White House, Frances Arnold is the advisor of the Joe Biden, the president, and also 20 editor-in-chief for the journals, and also many rising stars and big name, including, I think 22 national academy members already spoke or will be speaking. Also, the last thing, this is my advertisement. So if you are interested in microbiota for bioproduction, the deadline for the submission, the paper to biotechnology for biofuels and bioproduct, the deadline is the March 31st. And then another paper, a journal, is basically new biotechnology. I am basically served as editor-in-chief. So if you are having any manuscript that's good quality, you could submit that to this journal, and I'm basically handling this paper submission as well. And then the last thing is plastic problem. If you are interested in submitting plastic papers, I'm kind of currently editor for this specific topic with my colleague Mike, and then the deadline is on April 30th this year. With that, I want to thank all my student and postdoc, and also my two advisors, Christella Preder and Chris Voigt at MIT, and my collaborators, and also all the funding source right now. And then I'm happy. Thank you for your attention, and I'm happy to answer any question you may have. Thank you.
Okay, thank you so much for the talk, Professor Moon. So does anyone have any questions? We probably only have time for a couple before the award ceremony. Michael, it looks like you have a question.
Yeah, thank you so much, Alex. Thank you so much, Prof. Tae Seok Moon, for the wonderful presentation about various aspects of synthetic biology. So mine still goes to the aspect of biosensing. Among the various challenges that you listed down, I could just imagine how you could overcome the challenge of signal amplification in biosensing, so that even the minute concentration of the unlight, the signal that it produces, at least it can be amplified, because that still proves to be a challenge. Thank you.
Yeah, so the signal is always the challenge. So we had the problem when we first apply some literature reported sensor, and then their sensor only giving us twofold increase in terms of signal. So at the end of the day, we decide not to pursue further for that sensor, and then rather reinvent it using new mechanism. So that is the big problem, because you need to have at least the bigger signal than noise. And then that is something you need to play with by using some protein engineering. That could be some error from PCR-based, directed evolution or saturation mutagenesis. In that way, you could potentially improve the sensitivity and also fold change. Does it answer your question?
Yeah, yes, it does answer my question.
Okay.
Maybe if in the journal you have like certain articles that have tried to address the challenge that you could refer us to.
No, what is your question again?
It's about improving signal amplification.
Yeah, to improve the signal amplification, I mean, basically you need to, depends on what kind of sensor you're having. So there are multiple different ways you could change, including you could change the protein, sequence, targeting some, you could use some structure prediction algorithm. Let's say your one amino acid changed to another amino acid, you may see better signal. And the other way, people typically use is change the expression level of the sensor. In that, by changing promoter strength or ribosome binding site strength. In that way, you may have the better dynamic range. And then you could also use some completely different category of sensor rather than just stick with the one sensor system. In that way, you potentially have the better sensor, because nature has lots of different resources you could use, and then but one sensor is not necessarily good enough, but the other nature sensor, if you engineer right, you may have the better sensor system.
Yeah, thank you so much.
Sure. Any other question, or we need to move on, right?
Yeah, I think now we will probably move
Sure.
on to the award ceremony.
Yeah.
Maybe I see some people are still trickling in, so we'll just wait 1 second. But
Yeah, I
in
can
just
answer by typing, in the chat. If you have a question, you could send me email, or you could, chat, use chat, I will happy to answer.